He's also involved in the French Polar Institute, which I'll let him tell you more about, I imagine, as part of his talk. Vincent's background is he got his undergraduate degree at the University of Caen, and then he has a master's and PhD from the University of Strasbourg, and all in France. His research covers it's an integrative research program. As you'll see today, he's interested in topics ranging from social stress to the evolution of social signals and life history trade-offs and kin selection. And he's approached this work through both manipulative and more observational studies, and mostly in two taxonomic groups, the ground squirrels in Canada, which we'll be talking about, and then also king penguins. His work has been funded by a number of institutions. Most recently, he has a joint grant, from what I understand, from the University of Strasbourg and the CRNS in France. Vincent's been prolific in his publications. He's published more than 60 publications in his career in journals, excellent journals ranging from Nature to many of the behavior journals, even some of the regional journals that are important: Hormones and Behavior, ProcB, and Tree. And so with that, I'm really happy to have Vincent join us for the last find of the fall time. He is the title of his talk will be Social Stress and its Adaptive Value in Colonial Mammals and Birds. And Vincent, just to let you know, we are getting some on and off. I don't know if you're hearing it as well, but the recording, we're having some trouble recording to YouTube. I hope that doesn't interrupt you at all, but I'll do my best to keep it quiet. But thanks, Vincent, for joining us. And I'll turn it over to you now.
Okay, well, thank you very much, Lauren, for this great introduction. That was very nice. And I would like to thank all the hosts of the Find Seminar series for the excellent work that you guys have been doing. So Karsten, Adriana, Lauren, Eduardo, but also, of course, the backup team, Facundo, Jingyu, and Madison. And it's really an honor to be part of this amazing series. I must say, I usually don't. I'm usually not able to partake in them live because it kind of coincides with the time I need to go and pick up my daughter. But I've been watching them online, and they're really exciting events. So thank you for putting all those on. Can you guys see my screen just to be sure?
Yes, we can.
Okay. And so as Lauren mentioned today, I'd like to share a little bit of the research we've been doing both in the northern and the southern hemispheres. So I was quite pleased to see a few of my collaborators in the audience, so they can keep me straight if ever I'm talking nonsense. But basically today I'd like to focus on the study of social stress and its adaptive value in colonial mammals and birds. And this will be, I guess, no surprise to this audience, but group living is a fine balance that comes with a number of advantages and constraints. So the advantages that you all know, of course, are anti-predation, the ease of finding mates or resources, and across all of these advantages, you kind of have the opposite. If you have large groups that avoid you that allow you to deal with predators, you also become more conspicuous and so are more prone to predation. Looking for mates also means that you're competing for mates. You might share parasites in large groups, and both sides of this balance are kind of there's something that kind of pops out, and it's the management of stress levels. On one side, you're alleviating stress; on the other, you're accentuating it. And so, my research, together with my collaborators, has been interested in how social interactions affect individual phenotypes, and more specifically, behavior and stress physiology. But also, if you turn that and flip it around, how do individual physiology, how does that shape social behavior? And really, I've been trying to think about social environments as kind of this balance on the individual fitness axis, with the idea that beyond a certain threshold in social group size or composition, above that threshold, you might intensify, say, for instance, competition, or parasites, and all that stress is going to lead to social stress. But below that threshold, actually, the composition of your social group. So, for instance, your number of cooperators is going to allow you to buffer a certain number of environmental stressors, and that's the social buffer hypothesis. So you kind of have an optimal group composition and size where basically you're maximizing fitness, and that's how I've been thinking about social environments. And so I'd like to start by, so this is going to be a bit of a joint talk. I'm going to talk a little bit about ground squirrels, a little bit about king penguins. So bear with me. I know you might not see an immediate link between the two. And I'm going to start with the ground squirrels. So we've been studying ground squirrels thanks to the amazing early insights of Jan Murray, who started a long-term study on this species, and then there was followed up by Steve Dobson in northern Canada. And so we're working basically in the foothills of the Rocky Mountains on a ground squirrel population that we monitor every year. They're a social mammal with a short active season. They hibernate roughly 8 months of the year. And during their short active season, they have a very intense social life. They have a matrilineal society, so all the females cluster together in the same colonies. The males usually immigrate when they're sub-adults. They have repeated social interactions and social groups over the years. They can live fairly long. The females usually reproduce for the first time when they're 2 years old, but the eldest breeding female we had in our population was 14 years old. The females are going to raise a litter that's on average 3 offspring, can go up to 6. And yeah, we've been studying this population since 1992. And the first thing I was interested in when I started working with Steve and Jan was whether kin provided a fitness benefit to those animals, and whether that could explain the evolution of matrilineal societies. And so the first thing we did was go back into the long-term data and look at the records of female reproduction in genealogies from 1992 to 2010. And from that data, we were able to reconstruct individual fitness measures over lifetime for all those females that had lived their entire lifetimes and died. So maybe you're familiar with this type of analysis. It's basically a population matrix modeling, which we applied to the scale of an individual. So here in this matrix, you have on the top line here, you have basically the number of reproductions an individual female went through, and this is basically her number of offspring. You divide that number by 2 because only half of the genome is given by the mother, the other part being given by the father. So this would be 3 offspring, 3 offspring, 3 offspring, and 2 offspring. And of course, each year an individual survives, she gets a 1 until she dies. And from these matrices, you can, much like you would do population growth rates, you can get the dominant eigenvalue, which basically gives you an idea of the growth rate of the genotype. And so we used this method, which we corrected for population density, population growth rates, to ask whether individuals that had a lot of kin around them had higher fitness than individuals that had a few kin around them. And what we did was basically, I'm going to kind of skip through the details. That we did the path analysis where we looked at different traits which seemed important to us, such as the timing of emergence, individual structural size, or body condition, and we looked at basically how kin. This is all going awry. Okay, we looked at how kin affected individual fitness. And whether this was an effect that went through the number of offspring they produced, so that's litter size at weaning at the top, or whether this went through individual lifespan. And what we found was an indirect effect of the number of kin that you had in around you on fitness through the litter size they produced. So individuals that had more kin basically produced more offspring over their lifetime. And when we looked at this, that's the graph you have to your right. When we looked at how individuals clustered in terms of their spatial position, were they located their individual nest burrows where they raised the offspring in their environment, what we observed was that kin tended to cluster closer together. So this is the distance on the x-axis, the distance from an individual to the nest burrow of other females, other natal kin in the environment. And what you can see is that most kin cluster within a 30-meter range. So they really tend to cluster together. So individual females, ground squirrels that have more kin produce larger litters. But why? So we had two hypotheses there. Oh, I see part of that. Sorry, that sentence skipped. One of our hypotheses was that kin provided a safer climate to raise offspring, so less aggression and probably a safer climate against infanticide. The less aggression was already suggested in earlier studies. We also had the hypothesis that maybe females that had more kin were able to use different life history strategies and to allocate energy differently to major functions. So to look at how aggression varied between individuals that had a lot of kin or not, we did a social network analysis of aggression over a couple of seasons in this species. And what you have here is the social network we came up with, each individual triangle being a male, each individual circle being a female, and the blue circles being females that were breeding, the gray circles being females that were non-reproductive over this period. And each arrow represents an aggression from one individual to another, and the width of the arrows being the number of aggressions that are exchanged. The number you see within the symbols is the number of the mother. So basically, symbols sharing the same number had the same mother. So here you would have. Can you see my pointer or my yeah? Okay. I'm going to assume you can, but to the bottom of the graph, you can see basically 3 sisters and 2 brothers, just to give you an idea. And when we extracted the data from the social network, there's a few things that came out. So we calculated a parameter that's called strength, which is basically weighted aggressiveness. And what we found was that, so you can separate this into in strength and out strength. So in is the aggressiveness an animal would receive, and out is the aggression that an animal would emit towards someone else. And what we observed is that this was, of course, related to age. So the older an individual got, the less aggression it received. But the older an individual got, the more it emitted, up to a point where aggression decreased again. So as you can see here, you have one individual this 14-year-old female I was talking about. That was the longest lived female we had. When we removed this from the analysis, we basically got similar curves. They weren't as marked, but basically the patterns were the same. And when we looked at the mean number of aggressions that individuals elicited towards kin or non-kin, what we found was a strong difference between what you were dishing out basically to your kin or to non-kin individuals, and females were much more aggressive towards non-kin individuals, basically. So it does seem that breeding as a matriline provides you with benefits in terms of aggression. You're less aggressive towards family, and that probably is an advantage in terms of reducing infanticide, which occurs in this species. But what are the consequences of this on energy allocation strategies? So for this, what we did is we went back in the data and we divided energy allocation in terms of mass that the individual gained over a season versus what the individual expended in reproduction. So basically here on the graph, what you have is the litter mass that's produced over a season. So how much an individual invests into her reproduction, and this is how much an individual invests into overall, so into her reproduction, but also into her soma. And so if you look at the residuals of this, of course, you've got a very strong correlation between both. But if you look at the residuals, you're then able to separate females that allocate more mass to reproduction or females that allocate more mass to soma. And when you look at how that allocation, so this is the residual, anything that's above the black line here is more allocation to reproduction, and anything that's below the black line is more allocation to soma. if you look at how that affects, how that changes with the number of co-breeding kin that you have in the community, what you can see is a positive relationship with basically especially a strong shift from 0 kin to 1 kin. So as soon as you have kin in the community, you're basically able to allocate more into your reproduction than you are into your somatic effort. This was especially interesting because when we went through the previous models that we had, where we had a positive relationship between kin numbers and litter size, if you added in the energy component, which is what you see above, basically the effect of kin numbers became much less, which meant that basically the number of the effect of kin on litter size was especially going through energy allocation processes. So kin appeared to provide fitness benefits through lower aggression, but especially through changes in energy allocation processes. And so what that kind of brought us to think about was whether kin were acting as a social buffer to stress in this species. And to answer this, we had a dual approach. First, we just kind of went out there in a very life history type approach, and we worked over 2 years on 3 populations of ground squirrels, and we sampled. Sorry, I need to move this because I've got something in my screen. Sorry. Okay. And what we did is during the reproductive season, specifically during lactation, we sampled feces from females in the 3 populations, and within the feces we were able to measure fecal cortisol metabolites. So basically, those are the breakdown products of cortisol that is expressed at baseline levels in the animals. And what cortisol gives you is an index of how solicited your metabolism is, and basically it's used as an index of stress in a lot of species. And so we had an index of baseline stress throughout our different populations. And what we then did is that we looked at how those baseline stress levels changed with the number of kin that female had, co-breeding kin they had in the population. And this is the relationship we find. And so we were expecting a kind of a directional negative relationship. It's a little more complex than that. Actually, we find that kind of like what we found for energy allocation just before, when you shift from 0 co-breeding kin to 1 co-breeding kin, your baseline stress levels seem to decrease a little bit, but then they re-increase again when you have more kin in the community. So it turns out that family seems to be beneficial, but maybe not too much. So we kind of decided to back that up with an experiment, and this is the second thing we moved on to over a couple of years. And we thought about, okay, how do you manipulate the social environment? And we had a bunch of ideas, some of which weren't practical at all, such as trying to remove individuals from the population or trying to add individuals in the population, but that's logistically complicated, and besides, these individuals move about, so it's kind of hard to control. And then we just went back to the basics, and we went back to John Murray's brilliant original ideas, which was to manipulate not the number of individuals per se, but the indices of their presence. And so what we did was we simulated territorial invasion using scent marking. So these animals have scent glands just beneath the chin here, and they're and they spend their time marking their territories with those scent glands. And I don't know if you can see this little cube. So we used little plexiglass cubes, which we rubbed against the scent glands. And on this cube, if you can see, there's kind of a greasy streak mark that comes across the surface, and that remains on the cube for quite a while. And so we used those cubes, which we displayed around the nest burrow, so the burrow where females were raising their young. We displayed those cubes around the nest burrows of females. And we did this over a couple of days. The first day we displayed control cubes and let the animals habituate to them. And on day one, what we did was we started looking at the behavior of the animals once the control cubes were deployed. And 30 minutes later, we would take a blood sample. And 7 hours later, we would take a fecal sample in order to try and see the effects of the scent. So, of course, day one, there was no scent on the cubes. The effects of the scent on the stress axis. And then day two, we do the actual experiment, and then on the cubes we would either put the scent of a kin, the scent of a stranger, or the female's own scent as a control, and we would look at what happened on the behavior and the stress axis. So these are the results for the behavior. What you see here are basically multinomial linear mixed model effects, and how you read this is an effect from whether vigilance behavior increases versus the other behavior that's indicated afterwards when you expose the individual to different scents. What you see here is that when you look at, so any departure from one would be an increase if it's positive. Above one would be an increase in the odds of increasing vigilance versus foraging, or the odds of increasing vigilance versus other types of behavior. When you look at females that were exposed to stranger scents, basically there was an increase in vigilance compared to other behaviors, but to our surprise, it wasn't as marked as the odds of increasing vigilance behavior versus other types of behavior when you were looking at kin scents. So when females were exposed to the scent of a daughter or a mother or a sister, they would significantly increase their vigilance behavior. They would not increase their behavior when they were exposed to their own scent, which makes sense and shows that the experiment actually worked. The control's working. They're not reacting to their own odor, basically. Now, if we went, because we exposed the females within the kin scents to either the scent of their mother, their sisters, or the scent of daughters, and so we were able to actually go into a finer detail into the analysis, and this was really interesting because actually you could see that the females didn't increase their behavior, their vigilance behavior too much when they were exposed to the scent of a mother. They did, however, increase it quite a bit when they were exposed to the scent of a sister, and a little bit so also when they were exposed to the scent of a daughter. So basically, they didn't react too much to mothers, but they did react to their daughters and to their sisters when the scents were displayed on their territories. When we then went to look at what happened in terms of the stress hormones, in here I'm showing you the cortisol levels that were measured in the blood. What we found is that indeed, to our surprise, what seemed to cause the biggest increase in free cortisol levels was being exposed to the scent of a kin individual. So that was a 124% increase compared to the control situation where basically no scent was exposed. And we found actually no difference when individuals were exposed to strangers or to their own scents. And again, going down into a finer detail, and so smaller sample sizes here because we weren't able to collect as many samples on the daughter, sister, mother categories. But what's interesting here is that, remember earlier on I was saying that they reacted more strongly in vigilance to the scent of daughters or sisters, but actually when you look at the hormone levels, where they're reacting more strongly is in response to the scent of the mother. That's a 318% increase compared to no scent conditions. There is a bit of an increase for sisters, though not significant, and there's virtually no increase for daughters. So it's interesting because the behavior and the stress axis actually don't really tell you the same story. So when you look at the bigger picture, basically you can view this balance in group living and the effect of kin both on the positive and in the negative side. So kin seem to be able to help you reduce aggression, probably reduce infanticide, though we didn't test that per se. They allow you to increase energy allocation to reproduction. So in that sense, they're alleviating stress. But on the other side of the balance, it seems that mothers do cause some stress. They're probably dominant over younger females. Actually, we know this from behavioral observations. It's possible that part of the stress is related to local resource competition, probably for space. We don't think that foraging plays much of a role, at least at the scale of a season. And then other things come into play which might accentuate individual stress. So one of the questions we were interested following that was whether maternal stress had an effect on offspring phenotypes. So whether you could document transgenerational effects of maternal stress, because they have a quite short breeding season, and during lactation, a lot of those stress hormones can be passed to the offspring through the milk. So what we then did was we looked at the effects that mother phenotypes might have on offspring development, and we sampled individuals during lactation to measure their stress hormones, and we also measured individual mass during that period to see how that might affect the offspring they produced. And what we found, if you look at, so your y-axis here is mean offspring growth rate, so how much an offspring grow per day. What we found was relatively good relationships with maternal body condition. So all the variables are standardized here, with a maternal body condition and maternal free cortisol levels, which was interesting. So females with higher levels of cortisol or females in better body condition had offspring that grew faster over the growth period. But what was more interesting was that we found actually strong effects of cortisol levels on offspring survival. So mothers that had higher levels of cortisol during lactation had offspring that survived better to weaning. But even more interestingly, they had offspring that survived better the next hibernation. So remember here that the breeding period is relatively short, and then it's followed by an 8-month hibernation period. So maternal condition and stress levels here at weaning actually had an effect on how well the offspring survived the next 8 months during hibernation. So that kind of begs the question. I mean, Ben Dancer has been doing some exciting work on the red squirrels, also on the effects that maternal hormones have on offspring programming, and that kind of, so our data here is correlative, but it kind of begs the question as to whether similar effects might exist with maternal programming of offspring phenotypes depending on the social conditions. So of course, in our studies so far, what we've been doing is really looking at individual relationships above ground, and we've been doing this the classical way. We go out there, we use our binoculars, and we take notes. But what we're missing is everything that happens below ground, and this is something that a lot of people working on burrowing rodents are interested in. What takes place in the shadows, and so we've been slowly developing. So it's come with mixed success, because it's, well, technology, when you're developing technology, there's always drawbacks, but we've been developing a system which basically allows us to track individual interactions both above and below ground. And it's a project that's ongoing. We basically have small radio collars which record individual interactions. They also record behavioral acceleration on 3 axes. So we have an idea of what activity the animals are doing. And they also record body temperature and light, so we know when the animals are above ground or below ground. And the idea is to be able to reconstruct social networks both above and below ground and relate that to the life history of the animals and their physiology. I won't say more about this now, although I'm happy to answer questions. This is work that's still ongoing. So with that, I'm kind of going to make an abrupt change and jump into colonial seabirds. And focus on the king penguins. Now, this is a picture where you can see a lot of blood. And the reason I chose that picture is to illustrate the fact that colonial breeding is an interesting model, because it's basically very densely distributed territories with very little resources other than breeding sites. there's very high rates of aggression within those territories, and as you can see, the costs might be actually pretty elevated with breeding failure just linked with inter-individual aggression. And so I've been working in the Baie du Marin colony, which is on the Crozet archipelago, roughly 2 to 3,000 kilometers off the tip of South Africa. And we're extremely lucky in this colony because we have, so historically, when this research station was first built, it was built in the middle of the colony. Now they moved the research station up to the top of the hill. You can see it up there. And what's left in the colony now are our scientific facilities, where basically we can go and work on the birds, we can acquire samples, and we can immediately process them in labs in the middle of the colony. So we have roughly 22,000 breeding pairs in this colony. And so one of the first questions I was interested in when I started working on this system during my PhD was whether colonial breeding was actually stressful, given what I mentioned before. So one of the first things we did was look at oh, sorry, those dots have shifted. We tried to address this using heart rate in the species. So we analyzed heart rate using external heart rate loggers, which we were able, basically those watches that you use when you go and run. Well, we used those and we adapted them to the king penguins. And all those dots were supposed to be in the colony, and they're basically cameras. And what we did was we analyzed, we video recorded the penguins, and then we watched the videos, and we analyzed social aggressions on 20 birds. So we were able to analyze roughly 600 social aggressions, and at the same time we were monitoring heart rate. And so that's what heart rate looks like in a king penguin, and you can from these data what you can do is analyze the increase in heart rate in social situation conflicts. And what we did was we looked at two different situations. We analyzed heart rate when the birds were actively involved in an aggressive event, but we also analyzed heart rate when birds were bystanders, when they were simply watching the aggression and not actively engaged in it. We also tried to separate between physical attacks, so where there was physical contact between the birds, or threat interactions where basically the birds were threatening each other, but not with no actual physical attack. So the risk basically was very different between the two types of aggressions. So this is what we found. Here you're looking at a graph where you have heart rate increase from baseline. So it's how much it increases from your baseline value following an aggression. And what was kind of interesting for us, well, not surprisingly, attacks where you have physical contact, you have a heart rate that increases more than aggressions where you don't have any physical contact. But what was really interesting was that even in bystanders, so birds that were not directly involved in the social interactions, there were significant increases in heart rate above the resting baselines. So there was a marked heart rate response, which suggests that the animals were highly sensitive to their social environment. Okay, so the birds, we've established that the birds are sensitive to their social environment. That's maybe not so surprising. After all, it is a colonial species. But does this actually affect your baseline stress levels on the long term? So to do this, what we did was we used a slightly different approach. We went into areas of the colony which were of low social density, and we also went into areas of the colony where the density was actually really high, five times the low density. And there we caught birds, we caught them and sampled them really fast, under 3 minutes, to try and get baseline glucocorticoid levels. And we measured corticosterone, which is the main glucocorticoid in birds, in the bloodstream. And what we found was that regardless of the breeding stage of the animal, whether they were incubating eggs or whether they were already brooding young chicks, there was a substantial increase in baseline corticosterone levels when the animals were breeding at high density compared to when they were breeding at low density. And this was between 33 to 54% higher, so quite a substantial effect. So we've documented that the birds are sensitive to their social environment, that the baseline levels are actually higher when when density is higher. But this is kind of an egg and a chicken situation, because what we don't know is whether the baseline levels are higher because of the social environment, or whether individuals that have higher baseline stress levels actually prefer more dense environments. So to try and answer this, we approached it from a slightly different angle, and we used a full cross-fostering design where we looked at what was going to happen to chicks if you raised them, or if the chicks were born and raised in the similar environment, or whether they were born and raised in a different environment. And so we swapped eggs both within low density environments and within high density environments, and between both types of environments. So eggs from low density were given to high density pairs, and eggs from high density were given to low density pairs. And then we let the animals incubate their eggs. The eggs hatched, and at 10 days and at 105 days, which is basically when the chicks thermally emancipated before winter, we acquired mass and a blood sample to look at what was happening in terms of their physiology. This was a huge endeavor that took us 2 years to do, over 120 pairs. And it was actually a lot of work in the field. So what we found are the following results. So when you're looking at the adults, so the adults basically, when we caught the adults, we sampled them under 3 minutes, we had their baseline levels, and then we held them and held them for 30 minutes before we resampled them. And the idea behind that was to try and look at the activity of their stress axis, so how much they were able to mount a stress response. And what you can see here is that we actually have quite marked differences in the response when the birds are at low and at high density. The high density birds actually don't respond so strongly to our capture. There is an increase, of course, but it's definitely not as marked as the increase that you can observe in the low density breeding pairs. Now, if you look at what happens in the chicks, so bear with me, this is a little more complicated, but basically, well, it's not that complicated. It's basically, those are the HD is high density and the LD is low density. So the first black dot here is chicks that were born in HD and they were raised in HD, high density, sorry. Chicks that were born in high density but raised in low density, chicks that were born in low density but raised in high density, and chicks that remained in low density throughout. And what's interesting, if you look at those black dots, is that if you take chicks that were either from high density or remained at high density, or chicks that were born at low density and placed into high density, what you see is that you basically have a dampening of the stress response. So when you change the chicks basically to high density environment, they tend to have lower responses to capture. So the stress axis seems to be pretty flexible in the regards that it's reacting to also how the social environment you're being raised in. We also found effects on weight, and what was especially marked was basically that your individuals that were in low density and that remained in a low density environment were those that had weight gains that were the lowest. So basically, it didn't really pay off being in a low density environment. And this was especially marked when we looked at survival probability of those chicks. So we continued monitoring those chicks over winter. And what you see here, so the black curve is the chicks that were born in low density and that remained in low density. And those are the ones that have the lowest survival prospects. But what's interesting is if you take the chicks that are, if you take the chicks that are in low density, but they were fostered and raised in high density, and that's the orange curve, you can see that actually they have the best survival prospects. So by moving the chicks from a low density environment to a high density environment, you're actually improving their survival. And that was probably the strongest result we had from this study. So basically, high social density does seem to increase baseline stress levels. It seems to also have an effect on the stress response. The stress responses are lower at high density, and high social density environments seem to promote chick growth and survival. So what this kind of seems to point towards is whether it's the parents that raise their chicks in high social density environments are probably overall better parents than those that raise their chicks in low density social environments. So contrary to what we thought originally, actually breeding in a high density is probably reflective of good conditioned parents and better survival prospects, despite the fact that stress responses are higher. And you can see the background there. That's a small snippet of the colony. So you can imagine how socially solicited those birds are throughout their cycle. So I'm going to stop there. I hope I wasn't too long. Just going to finish with a few acknowledgements. Of course, all our work on the penguins is funded through the French Polar Institute to provide us with the logistics to get down to those field sites and a substantial financial help to run those studies. I need to thank the AXA Research Fund and the Vincent Foundation that funded those studies. And of course, all my collaborators with whom I wouldn't be able to do the work without. So René Groscola, Jean-Patrice Robin, Pierre Bise, Claire Serrault, and Antoine Stier especially, but also a bunch of I mean bunch in the most positive way of students and field assistants, too numerous to list. And for the ground squirrel part of the research, of course, this has been funded by numerous grants of the National Science and Engineering Research Council of Canada, the National Science Foundation of the United States, grants from France, from the University of Strasbourg and the CNRS, the Foundation Vincent, and of course we thank the Alberta Parks and Government of Alberta for allowing us to conduct the studies there, and the Geoscience Institute from the University of Calgary. As I mentioned during the talk, the ground squirrel long-term study was initiated by Jan Murray and run for a long time also by Steve Dobson, and so I'm truly indebted to both of them for implementing the study and for allowing me to continue. And I also numerous collaborators, Rudy Boonstra, which was central in running the hormone analysis with students, Phoebe Edwards, that I also saw in the audience, and Peter Neuhaus, Claire Serrault, and a whole host of students and colleagues. Thank you very much, and I'll be happy to answer any of your questions.
Thank you, Vincent. That was really fascinating. It's cool to see it all come together, as I talked about beforehand. And I really appreciate your talk there, especially how you integrated behavior and physiology. So at this time, it's an opportunity for us to ask questions. And if you're new to the Fine, or if you just need a reminder, please post a question mark in the chat if you have a question, and I will relay that to Vincent. If you are watching in YouTube, please post your full question, and I can watch YouTube and I will relay that to the speaker as well. We really want to encourage the more students, postdocs, to please ask questions. This is a great opportunity for you to explore this research. So if you're a student, just or postdoc, put an S next to your name and I'll prioritize you in the queue. And so with that, if you have questions, please just put that question mark in the chat and we'll get started. So I'll get it going because there's no questions yet. Sometimes it takes people to get going. So Vince, I had a question about the ground squirrel project. Have you looked at, or you've been able to look at the relationship between litter size and cortisol in these animals, or baseline cort?
Do you mean between the maternity? You mean the offspring's cortisol levels?
No, the mother's level. So do you have data? I don't know. Forgive me if you showed that. I'm multitasking with hosting here. So I'm thinking, do you know, do females with larger litters have higher cort?
Yes. Actually, I did. Maybe I went a little fast. Actually, I did show that.
Okay.
But so basically, what we found is a relationship between females that have higher cort levels during lactation. Well, actually, sorry, no, I didn't show that. My apologies, I'm mixing up stuff. No, what we didn't look at litter size. Sorry, we looked at offspring growth rates. We focused specifically on growth rates.
Okay.
But I haven't looked at a litter size per se. So I can't
I
answer.
think that because you were talking about energy allocation, that led me to that question. That
Yeah,
females in good condition can allocate more energy to reproduction. They may actually have larger litters, and that can correlate positively with court because court's necessary to sustain lactation, right?
absolutely. Actually, that's a very good suggestion. Something we ought to look at. When we did this study, we really went down the route of growth rates. The idea being that a few papers had kind of documented maternal effects on growth rates. So actually, that's kind of what I focused on first. But yeah, your comment makes total sense. I should investigate that as well. Yeah.
Well, I can refer you to we have a paper on that with the Degus.
Okay.
It's based on Francis Bonnier's review article in Tree in the late 2009 or something. But I can get you that if you want. That I think that'd be really cool given the data set you have.
Yeah, absolutely. Thank
Great.
you.
I'll come back if I have more questions later because I want to get to other folks. So I see Anna Marie.
Yeah. Hi, Vincent. Thanks for a great talk. I'm Anna Marie. I'm a postdoc at the Universidad Católica in Chile. I have a question also about the ground squirrels. I was, of course, there in 2012, 2013, and I remember 2013 there was a big flood.
Yeah.
And I also saw that you saw some of the data also showed the result of 2013, 2014 with stress and everything. Did you see changes after the flood and what happened with the population?
Yeah, that's an excellent question. And my short answer to you is that we didn't have that many ground squirrels that were sampled after the flood. And it was actually something that Rudy mentioned at the time. That it'd be interesting to do a study on how they reacted to an extreme event. But at the time, it was just too complicated. I mean, it was really, the females were super jumpy. We lost all the litters for one of the populations, so it was really complicated, and I basically gave up on trying to trap them. But what we did do is that last couple of years, I had a student work on this question, and what she looked at were punctual events. So she looked at late snowstorms, and she looked at some rainfalls, and she sampled. So it was actually pretty insightful. What she did was she looked at weather patterns that were coming over the Rockies, and she anticipated when there were going to be major rainfalls and major snowfalls. And she went out before those events, and she looked at the behavior of the ground squirrels, and then she took fecal samples from them, and then she went out after the events, and she did the same thing. And what she showed is that and she went out during the events she had me go out during the events to look at nothing, but she needed to quantify nothingness, so we quantified the ground squirrels not being there, and what she did show this was that when the events came through, so the snowstorms or the rainstorms, the ground squirrels went below ground. That we already knew, but now we had some quantitative data on it. But what was interesting was that the stress axis actually was downregulated. We were expecting to see higher stress after the events, but actually it was the contrary. The stress hormones all dropped. And our hypothesis behind that was that basically they were not entering torpor per se, but they were metabolically downregulating the whole system, so that they were avoiding to basically tap into their glucose and their other reserves. Because I mean, stress hormones, it's a bit of a we use stress a bit lightly, a bit loosely, I guess, in the vocabulary. It's glucocorticoid hormones, which are before all the regulators of the glucose metabolism, and they're central effectors of other functions in the body. So that's what we think was going on.
Interesting. Thanks.
Okay, so our next question is from Carson. I think I'm gonna read his question for him. His question is there any competition with other any other small mammals on the field site? For example, some murid rodents.
Not that I know of. Maybe, yeah, not that I've documented. What we do know is that there are ears of high vole or low vole populations, and that we've seen. As to documenting competition per se, I couldn't really say that there is. What I have seen on the occasional, actually, I saw this once. The first year I was out there, I saw a ground squirrel eating a vole, running around with a little vole in its mouth. Whether that ground squirrel actually predated the vole or just picked it up, I don't know. But I mean, we don't have any data actually that would allow us right now to test the hypothesis that there's competition per se. I guess in terms of resources, that's what Karsten's thinking. Maybe Steve and Jan can weigh in, but to my knowledge, I don't think we have any data that allows to say that there is.
Okay. I imagine Carsten has maybe a follow-up, but he does right here. He says in bush karoo rats, neighbors might be using useful in chasing striped mice away. So it's
yeah, I don't think that happens too much in our system.
Yeah, I don't think there are very many other small mammals on those sites. In the nearby trees, likely more, but we've had a couple of occasions of other occasions as well of squirrels eating voles, but they're not very, in my recollection, they're not very abundant on those meadows.
great.
Thank you, John.
All right, so Vincent, just so you know, before I get to the next question, I popped the 2 references in the chat.
I saw them. Thank you very much.
Helpful to you. The next question goes to Zulema, please.
Yes, hi, Zulema Tang Martinez, retired from the University of Missouri in St. Louis. I'm wondering about the penguins and the high density populations, the chicks doing better under those conditions. And I was wondering whether there could be any characteristics, I don't want to say resources, but any characteristics of the high density habitats that account for this. And I know that you, if I understood you correctly, you concluded that the parents were higher quality in those areas, in those nesting areas. But I wonder if there might be something about those particular areas that are superior, whether it's predation or any other characteristic that would result in those areas, those nesting sites, essentially being superior to the low density and therefore attracting more penguins so that you get high density areas.
Yes. Thank you for that question. That's very insightful. Actually, the answer is yes, there is. But I think it's a mix of things. So the traditional view of a king penguin breeding area quality is indeed related to predation, where central areas are thought. It's basically Hamilton. Central areas are thought to be higher quality because they detract predators, and that is certainly the case in what I've seen in the field. The petrels, so the penguins on land are it's the chicks that are predated, not the adults, but they're mostly predated by giant petrels, which tend to land on the outskirts of the colony, and they mob the individuals that are on the outskirts until they give up their chicks. So definitely the central areas were would be of higher quality. Now, the difference is that here we didn't specifically focus on only central areas, but really on areas of the colony which were higher or lower density. There's other factors that come into play. We know that there is some heterogeneity in parasite loads, which in the case of this study were actually a little higher in the low density areas. But what we also know from the behavior studies that we do on the birds, and this is work that Steve Dobson has also been doing on the birds, is that so these birds have mutual mate choice. They both use the ornaments of the partner in order to select the best quality partner, basically. And one of the ornaments they use is they have this auricular feather patch around the ear, which is yellow. And the size of that feather patch is related to aggression. So the larger those feather patches are, the more aggressive the birds are. And actually, the birds with the largest feather patches are found in more central positions, which is kind of the evidence that is pointing us towards the idea that it's more individual quality. So it's more aggressive birds that are able to raise bigger chicks, more aggressive birds that have lower stress responses, which also kind of goes tail in tail with this the Köppen style idea of stress and behavior being linked. But I think what's happening is that there's it's all those different pressures together that are actually shaping the system. It's not just one of them.
Yeah, that's really interesting. And also what you just mentioned about the parasites is interesting, because I think the prediction would be that you would have higher parasite loads in higher density areas. Do you have any idea why the parasite loads are lower in the high density?
I do, or at least I have suspicions. Actually, so the parasites we're talking about are ectoparasites. They're ticks, Ixodes urae. And the low density areas are also areas where there's a little more rocks and a little more grass. And the ticks are found, especially the nymphs are found under the rocks, and the higher density areas are flatter areas with more sand. So I think it's habitat selection from the parasites actually that's driving the difference.
Yeah, great, thank you. Nice talk.
Thank you for your questions.
So I have a student question in the queue next, so I'm going to jump ahead to that. That's Julian.
Hello, I'm Julian Adam. I'm working I'm a student, doctoral student at the Instituto de Neuroetología in Mexico. I'm actually also working on how social behavior in spider monkeys is affecting glucocorticoid levels. So a really interesting talk. Thank you very much. I really love that you do like all the different models. Like you have behavioral observations and the sympathetic and the HPA references, like the different measures. So it's really interesting, and usually people cannot do that. So it's really interesting to see those all aligned. I have two questions actually. I have a question about the cube experiment that you did in the ground squirrels. And I was wondering, you just reported the blood samples, right, for the glucocorticoid levels, but you also took the fecal sample 7 hours later. Did they align with what the blood samples said?
Oh geez, I'm trying to remember now. I think they did. I think it wasn't as marked. Okay, I'm sounding terrible right now. I don't remember exactly. I think the signal wasn't as marked. Part of the problem is that the glucocorticoid levels you have in the feces, they're more integrative and it takes a longer amount of time. So there was a study that was done in this species to kind of validate the time that you needed to detect a signature in the feces if you had a stressor, and that was roughly 7 hours. The main problem we had is that when we went out again 7 hours later to capture the females, it was often in the middle of the afternoon, and the females were nowhere to be seen. So we had smaller samples.
Okay.
So yeah, we basically I believe the trends were the same. I need to go back in the paper to look at it exactly, but from my recollection, the patterns weren't as strong.
But that's what I'm referring to. You have all these different types of measures, and it's so nice that you can basically compare them to one another, right? And then I have a more global question, because several of your results were pointing towards higher glucocorticoid levels being adaptive, right? Which has been a critique lately in the literature, that we should look more at adaptive parts of the glucocorticoid levels and not jump to conclusions that it's always like fitness decreasing to have higher glucocorticoid levels. What would your take, after all the research that you've done, would your take be on how good of a measure glucocorticoids are to assess fitness or well-being or anything alike?
Well, actually, that's a very very good question, and I think that's still I would advocate basically to be a little more cautious and maybe humble in what we're doing. I don't think you can really get a view of, what an animal's experiencing in terms of stress if you look at one metric. That would be my message. So for sure, I think that, you've got to remember that glucocorticoids, the whole stress axis, HPA, I mean, it evolved for a reason, right? It evolved because it was adaptive, because it promoted survival in both acute and also chronic situations. And that's a lot of the work that's been coming out of North America has shown that. But the other thing is that glucocorticoids, they regulate approximately 10% of gene expression, which is humongous, and it regulates anything from immunity to glucose metabolism to mitochondrial expression. So from that regard, they're a very interesting physiological mechanism to concentrate on, because they have so wide-ranging effects. But the flip side of the coin is that it also means that it's the kind of short links that we do between glucocorticoids and stress are perhaps a little too quick. I think a lot of this comes from the biomedical literature that has heavily imposed on our way of thinking. And I think the way you come, and I mean, we've done the same in our papers. We've associated, I'm not saying that we do things differently. We associate glucocorticoids with stress because it's just, I guess it's been the standard for a long time. Although I'm trying to be more careful now when I write about this. But I think the way you come around this is you try to multiply the metrics you're looking at. So glucocorticoid hormones are one thing. You might want to consider individual immunity and see how that's panning out. You might want to consider oxidative stress. And when you have all those metrics, then you have a clearer picture of how much the animal or the organism is coping, and how much of that might be an adaptive or a consequence of an adaptive strategy, or a consequence of what the animal is dealing with.
Thank you. I agree with a lot of what you're saying. Actually, I think one of the problems also with calling it stress, which you did in your presentation a lot, is that it conflates also with the psychological idea of stress, and if we look exactly at your data that has all these different metrics, then I have the feeling that other things are stressful and other things are metabolically stressful, like that two different things can be one could be metabolically stressful and the other one psychologically stressful. And I think your data shows that pretty well. And it would be nice to have like this distinction a bit more to what is perceived by the individuals also,
Yeah, absolutely. I totally agree with that. I don't know if it helps, but we recently, this was one of my students recently reviewed the literature about this, and I can post the the link to the paper she wrote about this, and it's this idea of we got to move beyond of glucocorticoids per se in trying to assess the effects of the environment on vertebrate health. So that might be some interesting ideas in there.
Thank you very much. Sorry for the long question. Thank you very much.
Well, that's great. Thank you for your questions. Next question goes to Flavien. Did I say that right?
Hello. Yeah. Hello. Thank you for your talk. I'm a first year of master's degree in ethology in University of Paris. And I misunderstood what kind of data will you get from the MAMTag project? And I was wondering, how long did you start working on this project?
Thank you for your question. So the data you get, there's actually 3 sorts of data. The first is radio contact. So when 2 individuals come into contact, the caller is going to record the identity of the other caller. So each caller records the identity of one another. So that's the first type of data. The second type of data is acceleration. So it's a 3D accelerometer. So you have an idea of the acceleration of the body along 3 axes. And with that, our hope is that you can reconstitute behavior. And the third type of data is light. So there's a light sensor on the collar. And so it records red, green, and blue light. And with that, we're able to do 2 things. One thing that works for sure is to see whether the animal is below ground or above ground. Basically, when there's no light, the animal is below ground, or it's night. But the second thing we're hoping to be able to do with that is reconstruct what the animal is foraging on, the big vegetation types, because we're able to dissociate the different colors. Although I'm not sure it's going to work in the end, because we did a few trials last year, and it seems to be a bit complicated.
Okay.
And sorry, and your second part of your question was, we started working on this, and I didn't present results here because of the time frame, but we actually have some first results. We started working on this in 2019. Yeah, that's it. 2019, we did the first trial, and then COVID. And so we weren't able to get into the field for a couple of years, and then we started again after that. And what our first results show is that the collars are basically able to detect social associations that are within 3 to 5 meters maximum. So it's fairly close contacts between the squirrels. And the social networks that we were able to do from the logger data, they correlate pretty well. I think the correlation is like 0.7 or 0.8 with what we're able to observe when we focus just on the contacts that are above ground. So we've got a pretty good association there. And what was more interesting in this preliminary data is that we built the social network above ground and below ground, and the 2 networks correlate very strongly. So basically, the individuals seem to associate with similar individuals, or with the same individuals above ground and below ground, which makes sense because, I mean, we're talking about matrolines which share the same space, so it would make sense that they meet the same sort of settings, social settings.
Okay, thank you very much.
Thank you.
All right, so we have a comment from Clara. Claire, do you want to say it, or do you want me to read it from the chat? You can give me a...
No, I just wanted to display Clara B. Jones' retired social evolution. I just wanted to display those 2 comments. That's all.
Okay, well, yeah, thank you for those. I read them. I need to think about the LE effect. I know that's something that a lot of work has been done. especially actually, I believe in seabirds. Though I'm not too familiar with the work per se. And in terms of the stress literature, I think it's because there's been a lot of I think people have conflated a lot of terms. Within stress, they've kind of made stressor, stress response. And so, yes, I agree that in the ecological literature, often stress is defined as a perturbation that reduces fitness, but actually, stress isn't the perturbation per se, it's the entire thing. Stressor is the perturbation. And in the ecological literature, there's a lot of studies that show that some levels of stress decrease fitness and some actually increase fitness. And it's complicated because people have been defining it in so many different ways. They've also been using the terms eustress and distress. And so, I mean, I think the field has been muddied a lot by the way people have been defining and using it very loosely.
Thanks a lot. Good talk.
Thank you. Thank you for your comments.
I don't see any new questions in the chat or on YouTube, but Vincent, I had a question about the stress work. Are you still collecting data on that as part of the long-term study?
We are. So on which, well, actually on both. Yeah, we are on both systems.
Sorry, the squirrel. My bad. On the ground squirrels. Because I wonder, have you considered thinking about the ability to turn negative feedback on?
Yeah.
Because we've done that with the degus, and we found some interesting results where communal care impacts the ability of mothers to, we call them stress mothers. We gave them court tablets. This is Carolyn PhD work. And we see some interesting results. I posted another paper in the chat where communal care influences the negative feedback efficacy of the mothers. Might be because what I'm thinking, this conversation has gone towards like what's stress and how do we think about it in ecology, and I think that would be actually a cool, fine working group or something, discussion group for bringing Michael Romero or something to have talk with us, because I think when we think about the stress response and at different scales like you are, there's one more, which is the ability how negative feedback works.
Yeah, absolutely. No, I totally agree with you. So yeah, we haven't done that because the obvious way to do that is to experimentally go and go and activate the stress axis, or go and manipulate the environment, and then look at how stress responses might be dampened or not. So as far as giving them external court, I did think about doing that actually. What I was interested in was trying to understand whether if you had this adaptive transmission of court to the young, whether if you fed females court experimentally, whether you would see offspring that grew faster, and whether you could actually go down and measure increased levels of court in the milk, or whether you had effects that were also happening in utero. So I wanted to kind of dissociate those two. Then I thought kind of against it because I don't want to mess up too much with our long-term populations. Sure. And Steve was going like, oh no, what's he doing? So, I mean, we didn't go that far, but it would be interesting. Yeah.
I think we gave them dexamethanosone to stimulate the negative feedback.
Yeah.
Study.
Yeah. So well, that would be to inhibit the response, right? And then you would give them ACTH to jack it up.
Yeah.
Yeah.
I mean, the lead author, Carolyn Bauer, she's at Swarthmore now. She'd be a great person to talk to about if you pursue that. She's a really good colleague. Yeah,
thank you. Actually, I've been collaborating a lot with Rudy Boonstra, that's been doing this all over the Arctic species. I mean, they especially worked on Arctic ground squirrels and Arctic hares. And that's typically the kind of methods they use to measure it. We more went towards, I would say, maybe a less experimental approach. We run after the squirrels. We put traps out, we have our stopwatches on, and then we run like crazy. We try to sample them as fast as possible. And actually, so we were doing this based on Romero and Reed's, and others that say that like 3 minutes is kind of your goal line for for having, baseline levels before it increases. So last season we thought, oh, let's test this, so we caught squirrels and we sampled them at different times. So I think the most rapid was like under 3 minutes, and we sampled them up to 20 or 30 minutes, I think. And actually it was pretty surprising when we looked at the females during lactation. Their stress response only started to increase, I think, after 25 minutes or something like that. So, I mean, it increased from the start, but I mean, the levels weren't really significantly detectably different from baselines before. I can't remember. It's paper's out in review right now. It's 25 minutes, I think. In the males, it was slightly. So actually, they're very, 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 during that period, during lactation, they're very slow to respond to, in terms of hormones, they're very slow to respond to an external stressor.
Do you think that's because their baseline levels are already so jacked up, or
the baseline levels aren't that jacked up? No.
Okay.
So I think but I do think that the whole systems, I mean, they're at a stage where they're also investing very heavily into lactation. So I think the whole system's just kind of shut down and everything's geared toward reproduction and less towards survival, basically. I think that's what's happening. Which is interesting because they still respond, right? I mean, they still respond behaviorally. I'll send you the paper.
That'd be great. Thank you.
Yeah.
Julianne, you had a question.
Just a quick comment. I was thinking about what you were just saying, Lauren. Might that all be related to oxytocin having also a negative feedback on glucocorticoids? Because have you looked at oxytocin as well in all those?
No, I haven't, but that's a very good comment. It could well be the case, yes.
Because I found in the spider monkeys, I found very low glucocorticoid levels in lactating females, which for energy levels, you would think that's contradictive, but I think it's oxytocin related depression of glucocorticoids.
Yeah, absolutely. No, that's a very good point.
Steve, you got a question or a comment?
Yeah, just to follow up on that. I'm thinking mainly about the ground squirrels, but this might apply more broadly. It looks like some individuals have a lot more energy capital to spend, if you like. So Lauren was talking about looking at relationship to litter size in the ground squirrels, and I wonder if looking at as sort of an index, the mass that's invested into reproduction, and then the total investment in reproduction in self during lactation, there's more of a positive relationship between these things. And I wonder if the females in the best condition can't mount the greatest stress responses, because they do everything well. And the stress response is really about reacting to a predator, or in your case, a pseudo-predator. And they simply have more energy to put into everything they're doing. So they're mounting a more appropriate response. And it's actually the ones that mount a very low response that have less energy, maybe less mass invested in both reproduction and reproduction plus their own personal investment, the somatic investment. So this leads me to think that taking more of a multivariate approach to looking at the different glucocorticoid measurements in terms of base levels, and then also the amount of response that's mounted, looking at several variables at once, you might be able to tease some of this apart.
Yeah, thank you, Steve. That's a good comment. Although we did look at mass effects at the time of capture on the response, and age effects as well. I mean, that's kind of the variables we also controlled for, and there wasn't an indication of a substantial effect of those variables on how strong the response was mounted or not. So yeah, I also think what's something that we need to go into now is, we were working at a particular time of the cycle where, females are towards peak lactation. They're exporting a lot of energy in milk, and that may well be very different at the start of the season, where either they're still gestating and milk production hasn't started yet. So there's also what was previously mentioned by Julian on oxytocin. So that's probably not interfering as much, but also their energy allocation is probably very different at that time. And I think we did this. I'll make a little confession. Basically, we did this study because I wanted to know if I had to keep continuing running after the ground squirrels to sample them, or whether I could allow more time in the field, because it was just becoming hectic. So short answer is I still need to run, maybe not as fast. But I think what the study also showed is that this idea of going out and sampling an animal under 3 minutes and saying, oh, that's your baseline level, that might be true, but not always, even within the same species, because that's going to be very dependent at the moment where you're sampling it, and at the moment, at that particular moment, what are the energy allocation trade-offs that the animal is making.
I'm looking at the queue and I don't see any questions, but I have more, so I can, if you don't mind, I'll keep going.
Keep going.
I'm thinking about individual differences now. And I don't know where this is going to go because my brain is all over the place. But I'm looking at that figure with the number of close kin and FCM. So it was early in the talk with the ground squirrels, and it was like a curve where the FCMs actually went up with the number of close kin, and that made me think of that question: does having close kin around help you to produce more offspring? Is that correlated to cohort? But the other thing you notice is that, if I understand this figure correctly, the variability is much higher among females when you have 0, 1, and 2 kin around. Am I interpreting that figure correctly, that there's a lot more spread, and it could be a sample size thing?
Let me pull it up and look at it again.
Back at I can't tell.
The variability is higher. I mean, definitely, I wouldn't conject towards the higher end of the figure, where there is indeed a sample size that's lower.
But even still, with that variability, it's like, maybe coming back to what Steve was saying about a multivariate approach, are there covariates there? Like you've looked at body condition, but do you know anything about masculinization levels, or which is indexed by the anal-genital distance in the degus and some other rodents, that could potentially explain that variability?
That's a good question. Off the top of my head, no. I mean, other than the idea. So this is an idea that actually Tim Cluttenbrock brought up really early in one of the presentations we did on the kin effects. So how we showed that that females that had more kin basically had higher fitness over a lifetime, and one of the questions was were females with more kin just also selecting higher quality areas? Were they maintaining higher qualities, basically territorial areas? And so you could imagine a situation where on average that's the case, but you also have matrilineals that just fall out of luck, and so you have higher variability. But with that being said, we've run a rough analysis of this idea with Claire Sarrault, and we didn't find any evidence that was the case. So no, off the top of my head, I'm not really sure what would explain a change in variability with the number of kin you have in glucocorticoid levels. I'd need to think more about it. There's got to be something.
That'd be a really cool analysis to do if you can.
Yeah, yeah. I mean, I'd be happy to continue discussing this with you if you have ideas.
Sure, my mind tends to go to masculinization, and I'll plug a talk in the spring. Loretta Correa is going to be talking about that because we found that masculinization explains a lot of things in degus.
So you would measure like testosterone levels in the females, for instance.
We have done some of that, but even just looking at the anal genital distance, which you can use as an index of exposure in utero.
Okay.
And
so in the adults, you mean?
Adults, yeah.
Okay.
I can talk more about that when we're not.
Yeah, absolutely. Yeah, that sounds interesting. Because we are starting some work now on testosterone levels in the ground squirrels, and especially how testosterone changes throughout the cycle and how that's related to aggression both in males and females. But we're really just starting this work, so I don't really have any results to present.
And I think Jane would be great to talk to as well because she's got that friendly male